Assalamualaikum and a very good day I bid to everyone. Today, our group will be presenting you a very interesting topic due to cancer, which is breast cancer. Breast cancer is the most frequent malignant disease and the leading cause of cancer death among women in both economically developed and developing countries. Globally, 1.4 million new breast cancer cases are diagnosed each year, of whom approximately 1/3 die of the disease. Breast cancer is a disease in which certain cells in the breast become abnormal and multiply uncontrollably to form a tumor. Although breast cancer is much more common in women, this form of cancer can also develop in men. In both women and men, the most common form of breast cancer begins in cells lining the milk ducts. In women, cancer can develop in the glands that produce milk. Most men have little or no lobular tissue, so lobular cancer in men is very rare. In some cases, cancerous cells can invade surrounding breast tissue. In these cases, the condition is known as invasive breast cancer. Sometimes, tumors spread to other parts of the body. If breast cancer spreads, cancerous cells most often appear in the bones, liver, lungs, or brain. Tumors that begin at one side and then spread to other areas of the body are called metastatic cancers. A small percentage of all breast cancers cluster in families. These cancers are described as hereditary and are associated with inherited gene mutations. Hereditary breast cancers tend to develop earlier in life than non-inherited cases, and new tumors are more likely to develop in both breasts. There are several different types of breast cancer, including invasive ductal carcinoma and invasive lobular carcinoma. Firstly is infiltrating ductal carcinoma. It is starting in your milk ducts of your breast. This cancer breaks through the wall of your duct and spread to surrounding breast tissue, making up about 80% of all cases. This is the common type of breast cancer. The other type of breast cancer is invasive lobular carcinoma. This cancer forms in the lobules of your breast where breast milk production takes place and has spread to surrounding breast tissue. It accounts for 10 to 15% of breast cancers.
Firstly, I would like to talk about pathophysiology of breast cancer. Breast cancer basically expands the original includes blood streams or both. Metastatic breast cancers may affect lung, liver, bone, brain, and skin. The major risk factors for breast cancers are classified as reproductive, such as pregnancy-associated breast cancer, familial, such as inherited gene syndromes, and environmental and lifestyle, such as hormonal factors and radiation. Breast cancers are adenocarcinomas that begin in carcinoma in situ. Carcinoma in situ is an early-stage non-invasive epithelial cell growth in ducts and nodules. DCIS and LCIS are examples in situ cancers. DCIS refers to proliferative lesions that restrict to ducts and lobules without basement membrane invasion. DCIS affects women but may also affect males. LCIS arises from the terminal duct lobular unit and has a homogeneous appearance, preserving the lobular structure. Loss of tumor suppression's E-carnitine causes cells to proliferate in non-cohesive clusters. LCIS can be detected by biopsies and not by mammography and is more likely to be bilateral. Breast cancer has molecular, genetic, phenotypic, and pathologic heterogeneity. Tumor heterogeneity is caused by genetic, epigenetic, and microenvironmental effects. Different tumor sections shows great variation in growth rate, immunogenicity, metastasis, and therapeutic responses. Gene expression profiling identifies luminal A, luminal B, HER positive, Bessel-like, cloudy below, and normal breast subgroups. Subtypes have varied prognosis and therapies. Onco30X, ProSignal, and MammaPrint use gene expression patterns to stratify tumors. This information helps to tailor breast cancer therapy and decides whether women require aggressive, systematic treatment or careful observation for indolent tumors. The driver genes play key roles in breast cancer development and progression. Examples of key driver genes include HER2 and M1C, while examples of tumor suppressor genes include TP53 and BRCA1 and BRCA2. When a tumor clone is established, genomic instability may assist through the establishment of other subclones and contribute to both tumor progression and therapy resistance. Most of the time, the breast cancers generally manifest as a painless lump. However, the detectable lymph nodes in axilla, skin dimpling, nipple and skin retraction, nipple discharge, ulceration, reddish skin, and bone discomfort from metastasis are the further symptoms. So, surgery, radiation, chemotherapy, hormone treatment, and biological therapy are used to treat the breast cancer based on the extent of the cancer.
In breast cancer, genes involved are BRCA1 and BRCA2. BRCA1 can be found at the long arm Q of chromosome 17, Q21. Meanwhile, BRCA2 can be located at long arm Q chromosome 13, Q12, RT13. The most common cause of hereditary breast cancer is by inherited mutation in the BRCA1 or BRCA2 gene. In normal cell, these genes help to make proteins that can repair the damaged DNA. Each person has two copies that inherited from each parent, one from paternal and one from maternal. These genes are called the suppressor genes because they can develop into harmful mutation, which is a cancer, when they have certain changes. Moreover, a person who inherit mutation in BRCA1 and BRCA2 tends to develop cancer at a younger age compared who do not inherit. In addition, by age 70, BRCA1 carrier women have a slightly higher risk of develop breast cancer rather than BRCA2 carriers. However, in most cases, if inheritance is by only one parent, they would inherit normal copy of other parents. This is due to the embryos cannot develop harmful variant from each parent, as the mutated BRCA gene will not functioning in repairing DNA. Mutated version of these genes can lead to abnormal cell growth, which can lead to cancer.
Next, we move to the next part, which is the mode of inheritance for breast cancer. The majority of breast cancer cases are not caused by inherited genetic factors. These cancers are caused by somatic mutation in breast cells that occur over a person's lifetime and do not pass in families. However, about 5% to 10% of breast cancer cases are assumed to be hereditary, which means they are caused by gene alterations, commonly known as mutation, that are passed down from parent to kid. The way cancer risk is inherited in hereditary breast cancer is determined by the gene involved. For example, mutations in the BRCA1 and BRCA2 genes are inherited in an autosomal dominant pattern, which means that only one copy of the changed gene in each cell is enough to increase a person's risk of getting cancer. Although breast cancer is more frequent in women than in men, the altered gene can be passed down from either the mother or the father. The gene alterations that raise cancer risk in the other diseases listed above are similarly inherited in an autosomal dominant pattern. It is vital to highlight that people inherit a higher chance of developing cancer rather than the disease itself. Not everyone who inherits this gene mutation will get cancer.
Molecular diagnostic tests have the potential to not only provide patients with personalized diagnostic information, but also allow for specifically tailored treatment plans, thus limiting resistant non-response and toxicity. Certain molecular diagnostic tests can also provide prognostic information about cancer in its early stages, thereby determining whether aggressive early management is necessary. The Mama Print test is a molecular diagnostic tool that assesses a breast cancer patient's chance for tumor recurrence. The Mama Print uses a 70 gene signature that has been shown to have a independent prognostic value or clinical pathological risk assessment in patients with no negative breast cancer. The test requires a fresh sample obtained during a surgical biopsy to be sent to a laboratory in an RNA stabilizing solution for analysis. Another major prognostic marker that is currently recommended for the primary evaluation of invasive breast cancer is human epidermal growth factor receptor 2, which is also known as HER2. HER2 is an oncogene belonging to the EGF receptor family. Gene amplification of HER2 has been shown to occur in 10 to 40% of primary tumors, and HER2 protein's overexpression is found in almost 25% of breast cancers. Lastly, hormone receptors also used to diagnose breast cancer. The presence of estrogen receptors and progesterone receptors is currently a component of routine evaluation of breast cancer specimens. Estrogen receptors, first analyzed in breast cancer in the late 1950s, was the first molecular marker evaluated for the prognosis and therapy response for the breast cancer. Estrogen receptor status has been shown to have a significant predictive value on tumor response to hormone therapy in metastatic disease, as well as for the adjuvant therapy after local excision.
There are a lot of treatment for breast cancer. The first one is surgery, which is the removal of the tumor and some surrounding healthy tissue during operation. Lymph node removal surgery is the removal of 1 to 3 or more limbs.
Axillary lymph node dissection is removal of many lymph nodes from under the arm. Reconstructive surgery is a surgery to recreate a breast using either tissue taken from another part of the body or synthetic implants. A breast implant uses saline field or silicone gel field forms to reshape the breast. Next is radiation therapy, which is the use of high-energy X-ray or other particles to destroy cancer cells. There are several different types of radiation therapy. A radiation therapy regimen or schedule usually consists of a specific number of treatments given over a set period of time, such as 5 days a week for 3 to 6 weeks. Radiation therapy often helps lower the risk of recurrence in the breast. Next, chemotherapy is the use of drugs to destroy cancer cells, usually by keeping the cancer cell from growing, dividing, or making more cells. It may be given before surgery to shrink a large tumor, make surgery easier, and reduce the risk for recurrence. Targeted therapy is a treatment that targets the cancer-specific genes, proteins, or the tissue environment that contributes to cancer growth and survival. These treatments are focused and work differently than chemotherapy. This type of treatment blocks the growth and spread of cancer cells and limits damage to healthy cells.
In conclusion, breast cancer is the major cause of cancer death among women. Breast cancer is a condition in which specific cells in the breast become abnormal and grow uncontrolled to create a tumor. The gene involved determines how cancer risk is inherited in hereditary breast cancer. Cancer does not affect everyone who inherit this gene mutation. There are two important aspects in breast cancer prevention, which are early detection and risk reduction. Screening may identify early non-invasive cancer and allow treatment before they become invasive, or identify invasive cancer at an early treatable stage. But screening does not prevent cancer. Breast cancer prevention really must be understood as risk reduction. In extremely high-risk patients, such as those who have BRCA mutations, risk reduction may involve prophylactic surgical removal of the breast. For the average patient, lifestyle modifications such as diet, exercise, and weight loss may be easily recommended and have many other benefits. For patients who have an increased risk based on other factors, the hormone-blocking agents are used. When the breast cancer is detected early and hasn't spread to anywhere else in the body, the 5 years relative survival rate is 99%. That's why being proactive about your health through early detection is so important. That is all from us. Thank you everyone for your attention and have a nice day.